Good evening, everybody. We'll just give it another minute and we'll be starting shortly. Welcome, everyone, again to a very interesting and a very different kind of a presentation today in today's webinar. Before I introduce my speaker for the day, a little bit about SigTuple Technologies Private Limited. SigTuple is engaged in making pertinent solutions in pathology on an AI-based platform. It uses optics, robotics, and data science to create pathological solutions on an AI-based platform. And as you all know, we have our products, which is the AI 100 and the recent AS76 as well. We all know that morphology plays a very important role in bringing out diagnosis in hematopathology. And when we talk about morphology, we are talking about things like WBC morphology, we're talking about RBC morphology, and platelet counts and platelet morphologies as well. All of these are part of the essential things that go into, creating the impression for a peripheral blood smear. And with digital in place today, it has become so easy and convenient to look at the results with the image-based evidences provided with the AI as well. And you have fantastic platforms like ours, like the A76, where you can literally just zoom in, zoom out, pan across the entire slide, and arrive at a diagnosis. And even zoom in and zoom out into the whole slide scanner view as well. All of these things have definitely created a huge bound and a leap in the area of laboratory practice today in hematology, as well as in other fields of pathology as well, where basically there have been advantages to the pathologists in terms of turnaround time, the standardization, the reproducibility, the quality-driven reports that do come. And we all know that everything starts with morphology, and that is the cornerstone, that is the gold standard. And with image-based evidence, digital pathology has definitely come a long way. But coming to today's talk, while yes, we start from the foundation, which is morphology, we have with us today our speaker, Dr. Utso Guha Roy, who is affiliated to Udayam AI Labs. And his topic for today is going to be AI-driven minimal residual disease, MRD monitoring in leukemias. So that's how far AI has actually come. And we will understand from Dr. Roy today how beautifully AI can be used even in advanced fields like MRD monitoring in leukemias. Dr. Roy's interest is varied, as you can see: AI education and literacy and capacity building for clinicians. Digital pathology itself has been an area of expertise for Dr. Roy. A lot of notable achievements: the certified in AI medicine, postdoctorate certificate course in digital pathology, certificate course in evidence-based diabetes management, and basically an MD pathology from Kolkata Medical College. So the list goes on and on. He has a lot of papers and publications in multiple peer-reviewed public journals. He's been a leading speaker in different national and international conferences, seminars, and also a member of various prestigious associations like the Indian Medical Association, Society of Digital Pathology, etc. So without further ado, I would like to take a pause out over here, and I would like to welcome Dr. Roy, our speaker for today. Welcome, Dr. Roy, and we're very excited to have you here with us today and to hear your perspectives about AI and MRD in leukemia screening. Over to you.
Thank you, ma'am, for that excellent and very nice introduction. So, very good evening. I'm Dr. Utso Guha Roy, so I'm a consultant pathologist. So I'm kind of working in the intersection of pathology and artificial intelligence. So the title on your screen, that minimal residual disease detection and monitoring in leukemias, might sound like a narrow technical topic. So it's not. So what we are about to explore is in many ways the frontier where biology meets computation, in many ways where a single cell can determine a patient's fate, and where machine learning is fundamentally rewriting how we make those life and death decisions. So when we say a patient is in complete remission, generally we used to mean classically fewer than 5% blast in the bone marrow smear. So morphologically clear, congratulations, you are doing better. But now we know today that this macroscopic view is largely insufficient. So we are now hunting for the persistent malignant clones hiding completely below the threshold of a human eye in the microscope. So the consensus is brutally clear. The presence of even a single leukemic cell in 1 million healthy cells can spark a full-blown MRD. So that is why MRD is now the single most powerful independent predictor of relapse-free survival across ALL, AML, and CLL. It's absolute focal point of the modern leukemia care, surpassing even cytogenetics and other risk scores. So this is one paper that we will come again at the discussion. So this is the ELN David MRD Working Party paper published in 2025. So obviously we have our gold standard, that is the multi-parameter flow cytometry. So it is rapid, it is a relatively cost effective, and it's beautifully applicable across most disease types. So here it is what the story gets little uncomfortable. So for a modern day 10 to 12 parameter panel runs on a clinical sample, it generates around 3 to 4 millions of individual cellular events. So each tagged across a dozen fluorescent parameters. And the ELN 2025 guidelines now demand a sensitivity of 10 to the power -5. So it takes around 15 to 25 minutes per case. There's also interpretable variability, and sometimes it leads to delayed reports. And ultimately, the pathologist burnout. So in a high volume clinical trial setting or a high volume centers, or like a reference laboratory, this bottleneck does not just cause inconvenience, it's fundamentally limits the scale at where the precision medicine should actually work. So this is actually not the critique of our colleagues of ourselves. It is an honest acknowledgement that the data deluge has simply outgrown the human visual processing capacity. So now on the top of that bottleneck, one more biological adversary antigen escape. So the modern targeted therapy like blinatumomab attacks the CD19 or rituximab attacks CD19. The CAR T cells destroys whatever surface markers they are engineered for. And the leukemic cells are also not sitting passively. So after a therapeutic pressure, the residual cells sometimes downregulate the very surface antigens that our diagnostic anchors depend on. So it can render the standard sequential manual gating strategy functionally blind. To a manual analyst working on a conventional approach, that population may quietly slot into maybe a different group if antigen is lost and the complete remission may be declared. So, and this is precisely why the scenario where high dimensional AI analysis is not optional, it is essential. So coming to the starting, so the problem we know that the sensitivity of the human eye cannot always reliably work at a very high scale against a biological target that actively shape shifts. So how does our AI arsenal actually look like? So welcome to the automated pipelines. So automated MRD deployment has a dual kind of strategy approach. First, we can use the supervised learning algorithms like the random forest or XGBoost, where the AI learns by examples of massive datasets previously labeled by human experts. And second, we can also use the unsupervised learning models like UMAP and others. Think of UMAP as taking impossibly complex multi-dimensional topographic data and flattening it into a 2D subway map, metro railways. So the human eye can easily comprehend. It squashes that data into a visual map that human eye can comprehend, keeping the clusters of abnormal cells spatially separated from the normal ones. So coming to examples, actual examples. So one is CCADDS or CADAS pipeline. So this is validated for BALL and multiple myeloma. It's mostly developed from Mayo Clinic. I think they're using it there also. So the timeline that we are saying, like 15 to 20 minutes per analysis, comes down to 60 seconds. That is around 90% reduction in time. Then the raw data that around 4.1 million events, it is reduced by 95% to a much lower number. So all the while preserving the positive agreement, that is the preserving the MRD positive clusters. So, and also it's shown that when validated against a expert manual analysis, it achieved almost complete concordance with the MRD positive cases. So I can understand there could be a skepticism, like you're saying you are deleting 95% of the data. So does not delete the actual MRDs also. No. So the cadres actually uses a cluster informed density based downsampling. So it mathematically identifies the rare clusters 100% times, and while aggressively clearing the massive redundant volumes of benign background population. So it actually doesn't delete the needles, it reduces the haystack and keeping every needle. So the pathologist is handed a curated high signal landscape rather than 4 million or 5 million dots. So what are the base under the hood of the Keras pipeline? Actually, it has multiple points which actually addresses different failure points for, not failure, maybe chances of failure for manual analysis. First one is flow cut. So it's kind of a bouncer. It's autonomously strips out the spurious events like caused by bubbles, clogs, voltage shifts. So the analytical step before it begins, the dirty data is already gone. Second step is the PARC. So it applies a phenograph based clustering to map the distinct cell population from clean data. Step 3 is the density based downsampling, where, as I mentioned, the intelligently manages data volume while protecting our rare events. And step 4 is the UMAP, which projects the high dimensional marker data onto a 2-dimensional coordinate for the human eye to navigate easily. And lastly, there is the deep neural network classification, which isolates the malignant clones from normal subsets and achieving a F1 score of around 0.86%. So the result is a lean, high confidence MRD readout under 60 seconds. So actually how it does is that it uses difference from normal philosophy. So instead of trying to teach the AI every single possible variant of a leukemia, so which is mathematically impossible because we know how varied they are. So the AI is actually trained exclusively on the exhaustive atlas of normal bone marrows that we have. So by deeply understanding the baseline, the normal, the AI can assign an abundancy scale to the patient sample. It flags anything that statistically deviates from the healthy pattern, and the abnormal cells are left nowhere to hide. So this was about BLL and multiple myeloma. Coming to acute AML actually, so it's a kind of different computational challenge all day. So AML is not a single disease as per se, it is constellation of biologically diverse entities, and we know that residual cells of AML share a subtle shifting surface similarity with normal regenerative myeloid precursors. Could be monocytic lineage cell, could be granulocytic precursors, could be erythrocytic elements, depending on the class of AML. So for any standard algorithm or to even a fatigue human expert eye, a post-chemotherapy bone marrow in AML can sometimes look staggeringly like residual disease or vice versa. So here comes Magic Deer is a machine learning guided AML MRD analysis. So it was built specifically for this unique biological landscape. So this was the actual paper published. So it pairs exhibit classifier for single cell prediction with UMAP for population based visualization. So it combines the discrimination power along with interpretability. And the validation data was AURC was 0.97 across diverse AML types. And most importantly, it was 100% concurrence with the manual blast detection, establishing direct clinical equivalence with the expert hematologist reviews. Okay, coming to other models that are beyond Magic Deer. So Magic Deer is not an isolated achievement. The evidence-based, AI-assisted AML MRD classification is rapidly expanding. So we are having random forest classifiers have demonstrated robust classification performance with interpretable feature outputs. Even there are support vector mechanisms or light GBM variants have shown competitive performance in recent comparative studies. One paper in 2025 in Hematologica, it is particularly noted that it's demonstrated that machine learning approaches could support rapid AML MRD identification along with strong classification performance. That means that the algorithm can classify the MRD cells logically onto the hematopoietic maturation pathways. So AI is not just pattern recognizing and statistically, it is also reasoning in a way what a trained pathologist, hematopathologist or flow cytometrist can follow, verify and trust. So the question is no longer whether AI can classify these patterns, it is how safely we can reproduce this in our own setup and when they're available as open source or maybe a paid tool. And that is the clinical challenge at our current context in India mostly. One interesting case study that highlights the importance of AI analysis was antigen loss after blinatumomab therapy. So this is targets the actually CD19 cells. So after this, it does say after the therapy of blinatumomab, it has done its job almost very well. So the surviving leukemic cells were CD19 negative, and now the standard diagnostic anchors has been erased. So conventional sequence getting can likely exclude this population from the B cell analysis totally and may be called a complete remission. So patient is discharged and the disease still present, still dangerous. So what the deep neural network does, actually it also looks towards all other dimensions of abnormalities into the cells. So I will go to the next slide and explain it more this way. So the manual gating, he will relies on the primary anchor, but AI sees the multidimensional aberrancy of the tumor cells. So it analyzes all the 12 markers or 10 markers, whatever is feasible for that tumor, and it will flag that leukemic cell based on the secondary markers also. So it doesn't need a single antigen positivity. It identifies a cell by its total abnormality that it has. So that way it can segregate these type of cells as they are not actually in remission, but actually these cells are persistent even after the chemotherapy is gone. Now shifting towards different modalities from flow cytometry to a qPCR or PCR for MRD detection, particularly for fusion transcript like BCR-ABL or NPM1 mutations. So they remain important for certain contexts. So the standard CQ or CT thresholding has an inherent structural limitation, I would say. So it reduces the continuous amplification to a single data point. So for example, in a highly diluted MRD samples, they have a near the limit detection. The late cycle amplification curves may fall below the fixed thresholds. And even in cases it has been seen like 4.3% cases has sometimes given as false negative. So for a high volume center malignancy in oncology setup, 4.35% false negative is a good number of people who can be damaged because of the missed relapse signal. So there are models like Spark which addresses this directly. It analyzes the entire amplification curve morphology, the shape, the slope, and inflection of the sigmoidal signals. Rather than single threshold, it analyzes the entire curve. And deep neural networks classify the raw RT-PCR curves by distinguishing this sigmoidal amplification. So AI is not just transforming flow cytometry, it is also augmenting the other domains by which MRD detection workflow works. Another area is the high resolution melting analysis, where AI is converting the subjective visual skill to an objective quantitative science. So it precisely analyzes the microscopic temperature shifts to identify specific point mutations, such as FLT3 in AML, eliminating the subjectivity of the visual interpretation by utilizing advanced frameworks that capture multimodal data structures. So for research consent settings, seeking an ultra-sensitive multiplex mutation detection without having an NGS, an AI-enhanced HRA may represent a clinically, economically possible way forward. So coming to Act 3, that is the global regulatory change that has happened in the last couple of years. So we come to that paper that I displayed in the first slide. So the European Leukemia Net released a landmark consensus document in 2025 where 57 specific recommendations developed through two-stage Delphi study process involved experts from around 29 international centers, and it had a 90% consensus, which is very high level of agreement in hematology domain. So the philosophical shift that happened in this paper was the MRD is no longer a binary positive or negative. So it's now more contextual, like 3 qualitative responses categories replace the old dichotomy. Now we have the optimal, the warning, and the high risk of treatment failure. Each carries different clinical action points, a different technological recommendation, and a different monitoring intensity. So, and for FLT3 ITD muted AML, specifically the guidelines now mandate ultra high NGS based MRD prior to allogenic stem cell transplantation. So the guidelines are no longer just telling us to measure MRD, they're telling us which technology to use and for which mutation at what exact point of the treatment timeline. So the era of contextual mutation specific MRD guided oncology is here, and obviously it can be enhanced with the use of AI. Another important area is where in the early 2026, FDA issued a landmark draft allowing MRD as a primary surrogate for accelerated approval of drugs. So this followed a unanimous 12:0 vote by the FDA's Oncology Drugs Advisory Committee. So why is this a fundamental disruption? Because the pharma companies can now no longer need to wait for 5 years to get a overall survival curves. They can now prove a drug's efficacy based on the MRD only and accelerate the speed at which a life-saving leukemia drugs can reach the market. Obviously, it has some economic, background also. It could have the big pharma and all those things. But this is a change that has happened in the FDA approval. So what actually led to this approval was 2 pivotal studies in phase 3. One is the Cepheus and one was the Benefit trial. So the Cepheus trial compared DVRD quadrupled with versus the standard triplet therapy, and it showed that the primary endpoint MRD negative complete response was around 60% versus 39%. So the sheer statistical power of MRD proof supported the FDA's frontline approval before traditional survival curves takes 5 years to mature. Even in the Benefit trial, proves that MRD testing reveals the biological truth sometimes the standard metrics miss. On surface, a 3-drug and a 4-drug therapy looked equally effective, but when the highly sensitive MRD testing was done at 18 months, 53% of patients on the 4-drug regimen were completely clear of malignancy, whereas only 26% on the 3-drug regime. So MRD is not just a safer test, it's a more deeper, more accurate measurement of the actual treatment success. And with the help of AI, that MRD detection becomes more transformed, more powerful. So obviously, we have seen the analytical power of AI. We have to go through the what is the risk and what are the realities. So the most appropriate clinical deployment of AI in MRD is the one that changes, what the expert does, not the one that removes the expert from the process. That is a critical distinction we always mention in using AI in clinical setting. So what the AI can handle is the pre-processing, the raw acquisition of data, the removing of artifact, the prioritizing of populations by aberrancy scoring, or visualizing complex multidimensional model data in interpretable maps. What the hematopathologist retains, the review of the AI flagged populations in full clinical context, integration with the morphology, genetics, and the treatment history, and final diagnostics and the clinical call, which is with his own accountability. So AI changes the pathologist's task from being a exhaustive searching for the tumor cells to an expert adjudication. So the cognitive load shifts from finding the abnormal cell to evaluate the AI's highlighted candidates against my own clinical knowledge. So that is fundamentally better use of our expert human judgment. So we are not being replaced, we are being elevated. There are few primary areas where AI can fail in cases of MRD. So first is the atypical immunophenotypes. Say clones with unusual genetic presentation. Example, a trisomy 12 in CLL is a well-documented example. These kind of variations, in surface marker expressions can confuse even a well-trained classifier. So the algorithm was not trained on every biological edge cases, and so in very rare cases, it can genuinely fail giving a proper diagnosis. Secondly, there could be acquisition artifacts like fluid instability or machine compensation error during the raw data acquisition. So as we mentioned before, the flow chart actually helps reducing DITs, but no artifact removal pipeline is 100% perfect. And lastly is the benign mimickers. So we know that extreme cases of regenerating bone marrows, the hematogons and the other cells in the after the post-intensive chemotherapy, they look very odd. So this can outsmart even the difference from normal philosophy that we learned a while before, and ultimately it can confuse the model. So acknowledging these limitations is not a weakness, it is the cornerstone of safe and clinical deployment. Another threat is of data drift. So what is it? AI model is not a static entity. So once deployed, it performs. We cannot expect it to perform as it was at the beginning. So it is trained on a specific reagents, specific flow cytometry, and specific configuration and specific patient population. And we all know these things keep on changing. So the model slightly starts to lose its accuracy without if it is properly not monitored and maintained. So real world post implementation monitoring showed there was a data drift around 2.9% in clinical cases. So this can lead to a missing of cases or error in the diagnosis. For an oncology setup in leukemia, 2.97 is a very big number. And mitigation is not optional. So it is operationally standard to have a continuous electrical quality monitoring, attribute acceptance sampling running in the background, flagging the real data drifts in real time, triggering any revalidation or calibration of the model whenever the performance falls outside the predefined bound. So the AI pipeline without a drift monitoring is more a liability than a tool. And as we always say, explainability is very important for AI in clinical practice. So no clinician or hematopathologist can base a life-altering clinical decision, but just because a tool said this is MRD positive or this is MRD negative, without giving any underlying reasoning. So this black box problem is a legal, ethical, and a clinical workflow problem. So explainable AI will solve this using tools like SHAP or LIME. So they generates real-time interpretable matrices and that the clinician can evaluate. For example, the feature importance heat map on your screen shows that the system weighs CD19 and CD34, 45, 56, everything in the classification decision, and the actionable insight may look like a cluster isolated due to CD19 plus correlation with apparent CD56 expression. So the physician can now say to a colleague or a patient, this AI flagged this population because of this specific immunophenotype deviation. I reviewed the reasoning and I agree. So this is the standard of clinical accountability our field requires, and XAI is how we achieve it. To manage this, the FDA and EMA has released a joint framework containing 10 core principles for good AI practices. The key pillars include continuous lifecycle management and risk-based oversight. Furthermore, trust requires transparency. So as I mentioned, explainable AI using tools that generate feature importance heat maps or other matrices is mandatory so that the clinician can understand exactly why an algorithm has flagged a specific diagnosis or a specific cell or cell cluster. So this is result project is from 2024 to 2028. So it unites 22 organizations to build a standardized, decentralized frameworks for AI diagnostics. So their mission is equity, ensuring that a patient in a small regional clinic receives the exact same quality and accuracy for AI-assisted testing as a patient in a Tier 1 academic center. So it proves that overcoming bias and drift requires global data sharing also and global collaborations also. And beyond this, and as a future, so throughout this presentation, we have framed that AI as a tool that can make a hematopathology or a tool that help the hematopathologist that they already do, like detecting a MRD. But I want to leave with a thought that we can push this further. So what if AI stops being merely our diagnostic assistant and begins to be our predictive intelligence? So the data emerging shows predictive models can already signal phenotypic resistance emergence around 3 to 4 months before a clinical manifestation. So we create a vital therapeutic window in which we can preemptively switch therapy before actually the relapse occurs. So imagine a situation where AI analyzes the residual clone, one cell in a million, and output is not just a present, but based on the specific multidimensional clone, this clone is predicted to exhibit resistant to current therapy within next 3 to 4 months. Then we start, stop treating the relapse reactively and start treating it more proactively. So leukemia as a preemptively can be managed like a chronic condition, not a cycle of constant remission and rescue. So that should be the ultimate paradigm this field should be building towards. So the future hematopathologist is not replaced by obviously by AI. So they are elevated by AI. AI will eliminate hours of manual getting, freeing the expert to do what no algorithm can replicate. Intricate morphology, genetics, treatment history, his clinical context and his clinical knowledge and domain, and the full weight of the medical judgment. So the pathologist becomes the commander of the digital marrow, interrogating and signing off on the AI prepared landscape with expert authority. So that is the vision, that is the destination and the journey. And thank you for patient listening. I look forward to your questions or any comments.
Thank you, Dr. Roy. That was indeed a wonderful presentation, so fresh and so different from the rest. AI has definitely come a long way ahead, right? And that's so much shown in your different slides. How, especially in MRD, can it really help in the future in predictive modeling, especially in determining equity, in underserved areas of population, where we can use the results of the AI along with management with the human mind as well, right? It's a collaboration of both in bringing up especially diagnosis in the difficult cases of MRD. It's
Especially.
definitely a very new concept which has come up. And it feels very good to know that the AI has actually dwelled so far, and not just in research, and probably will be coming soon in practical practice in pathology, in our day-to-day pathology reporting as well, especially in an era like flow cytometry and MRD, where still there is a lot of speculation and doubt in bringing about the right kind of diagnosis, whether we are getting the right kind of cells, whether the interpretation is right. It varies from person to person. Sometimes there's so much of subjectivity still involved in such areas, right? And having an AI-based platform for something like this definitely makes sense. It was an excellent presentation, and I definitely learned a lot from what the points that you have mentioned. And I leave it to the forum and the audience to see if they have any questions or any kind of feedback on the presentation today.
Thank you, ma'am, for your excellent words. Yeah, the field is very much evolving and is evolving very fast, I would say. Though majority of the tools are still in research phase, I think the SCADAS is being used in Mayo Clinic and majority are still in research phase. But
Right,
hopefully in the coming years, we will be seeing a lot of them being used and getting regulatory pass from the FDA and all others.
right. So we have a question in the chat. UMAP and TSNE are paid softwares for unsupervised learning or associated with any analysis software. This is by Dr. Asta Gupta.
Yeah. So, doctor, these are actually the machine learning models. These models are being developed on. So these are all machine learning terms and architectures. So on which these models are being trained on or developed on. So once the model will be coming to Indian market, hopefully someday, these all tools will be integrated within them, and most likely it won't be very much cheap. So initially it will have a cost to it, but yeah, I think with more exploration and maybe we can have our own Indian products. These days India missions are developing, giving a much more importance to health and AI. So yeah, we can develop something. Maybe in the near future we will having some models from India also, not we wait for the Mayo Clinic and other.
So we have Dr. Pratishta saying hello, sir. Dr. Pratishta, do you have a question as well for Dr. Roy? There is Dr. Indranil Chakraborty who has said, enriched by presentation. Thank you, Dr. Utso Guha Roy. So, yes, definitely a very enriching presentation, very enlightening at least. And there's so much of, take home message out over here that how we can exploit the role of AI, right? Whether it is basic reporting, whether it is on, advanced testing, all the way up to MRD. In fact, MRD is just being adopted in various parts of the country. It's still being adopted. It is tough, especially when it comes to AML MRD. ALL MRD is relatively easier. There is MRD for CML as well, and other hematological malignancies also, like multiple myeloma, etc. While we are getting used to that and getting our comfort zone in reporting MRDs, here comes AI again in the picture to make life even more easier for us. So it's
Yeah,
definitely a
hopefully it will reduce
welcoming,
the
yeah,
variability among ours and give
yeah,
at least a tool that we can use to verify our own judgment. And
absolutely.
yeah, And you
and
will
also
know
learn
better
from
than
it.
many of us because you are doing the hematopathology thing for a long time.
Yes, we are definitely into peripheral blood smear reporting and a lot of other parameters, of course. But definitely, AI in MRD is a very novel thing, I must say. And we really look forward to the time, Dr. Roy, when these things can happen in India under our own roofs as well, right? Even creating solutions for something like that. So, Dr. Asta Gupta has said, thanks. Is any such work happening in India, development of AI model in MRD, we'll love to collaborate. Uh,
Would love to collaborate also, but I think even if it's happening, it's very in a nascent stage, I
yeah,
think. But
yeah,
yeah, if I
at the
find
moment.
any, I will share to the SigTuple team
Yes,
and the links,
definitely,
and they
Dr.
can share.
Asta Gupta. We will keep all of you informed here in case if there is something on that front. And it'll be a great vision to take that forward for hematological malignancies, prognosis, predictive modeling, treatment, monitoring, all of that, right? Relapses, etc. I think it's a wonderful tool to have. Gaurav Mukherjee, AI-based MRD detection with treatment responses, retraining can produce an NLP, which can also help consultant hematopathologists. That's the comment given by him.
Yeah, yeah. True. I yeah. So I will share the presentation also to SigTuple team, and they can share it to the participants also if
Sure,
they
we
need.
will share. Anyway, the video of this will be shared, Dr. Roy.
Okay, then
The
it's
video
fine.
of
Yeah.
today's presentation will be shared with all of them who have attended the session. That's something that we do by default. So I think, one more round of going through this presentation itself will definitely, help us to put our thinking caps on to see, even in our day-to-day practice, whether we can actually start something in a very small stage. Everything starts from small steps, right?
Yeah, exactly.
Definitely a big
Absolutely.
eye opener. So Dr. Pratishtha is saying now, right now we're learning AML MRD in itself. That's so true. That's exactly what I'm saying. So people are still trying to get their comfort zone in understanding the concepts of MRD, especially AML MRD and other kinds of MRDs, right? We are still in that process right now. Adopting that and then, becoming a master of it and utilizing that in our day-to-day practice. So should we teach algorithms if we know too less? Dr. Roy,
Yeah.
your input on that, Dr. Pratishtha
Yeah,
is
I
saying.
think this is the where actually the human in the loop is important always.
Yes.
And
Yes.
yeah, and also the one thing is that there will be some kind of errors from in the training data also,
Mm-hmm.
because
That's
all of
right.
us not correct. So we train the models. So we maybe like annotate area in even in Hartinu say we're annotating low, ultra low these days. So it's not always that human data, human training data will be correct. So there will be some amount of errors going into the algorithm also. So that
Right.
is true. But actually, we need to upscale ourselves and then probably train those models and do it as unbiasedly as possible.
Right. So that's wonderful to know, Dr. Asta Gupta, that you're almost doing 60 to 70 AML MRD in a month. That's
That's
really
great. That's
amazing. Yeah, that's great. That's really amazing. So, if there is any chance of collaboration to do any kind of an AI-based platform, I think your center would definitely be very pertinent to collaborate with.
absolutely, yeah.
It was definitely a very enlightening presentation again, Dr. Roy, and we really do hope to have more of such talks on this platform as we did today. And we look forward to more interaction from the audience as well. If there are any more questions from you all, we'll definitely be happy to take that at this point of time.
I think on that question of is there anything being done at India? I
Hmm.
think what I'm finding is Tata Memorial is doing some work as expected because
Right. Right.
yeah, yeah. So they are something doing some work. I think it's not published. So they're doing some prepare ALL
Right.
consortium or something. So we can
Right.
explore those.
Right, right. So, Dr. Roy, don't you think that even a lot of the pre-analyticals also matter, right? Especially when it comes to flow cytometry. There's so much of pre-analytics also that goes into place, and the standardization of the flow cytometer in itself, checking the compensation and all of that in order to get the right
Everything,
kind of
yes, yes.
results. And
So
then
there are some
comes,
tools like
yeah, hmm,
flowcart and all those they have made
right,
into that
okay,
models to reduce the pre-analytical errors like bubbles, electrical
right,
charges, everything. But yeah,
right,
it is not
right.
foolproof, obviously, and I think in the
Hmm,
future there
hmm,
will be more better models that will come up.
Yeah. So to even have AI models on such things like compensation, areas where, a lot of pathologists actually struggle, right? To keep that standardization going on in the hardware and in the quality of the working of the tool in itself, leave alone the interpretation. Those also would be interesting areas to look at with a bit of an AI model which does quality check on the flow cytometer and the results which are obtained from the flow cytometer.
Yes, ma'am, absolutely. Because many are maybe focusing on the actual analysis. But as
Right.
we know in
Right.
lab medicine, the maximum errors happen in the pre-analytical phase, even in routine
Absolutely.
biochemistry to starting to MRD.
Right, right. We might even have AI-based controls in the future, to see
Yeah,
whether
could
these
have.
things
Yeah,
work. So there's such a long way to go and a lot of interesting ideas that can come up. And I think as pathologists, as doctors, we need to also put our heads together in where these things are of necessity and how swiftly we can actually work on bringing out such easy platforms, right? Tools that can be used by us. Finally, is created for our use at the end of the day. So Gaurav Mukherjee is saying, Dr. Utso, it's better to combine flow cytometry findings with fish results during the training of an LLM so as to reduce the black box effect. Yeah, I kind of agree with that question, but what's your take on that, Dr. Roy?
I think it could be a useful tool also. Yeah, and also may not be LLM, but maybe the training for the non-generative machine learning part,
Hmm.
and they can have some heat maps, saliency maps, or Shape line scores
Exactly.
or
That's
around them.
right. Right, right.
And without explainability, I think no clinician, no hematopathologist will sign off on a report by any tool.
Very true. Very true. Someday we'll have a collaborative diagnosis of morphology, flow cytometry, fish, and other kinds of tests as well, advanced tests, all of that put together where we have an AI model which actually gives us the
Yeah,
final diagnosis. I
yeah.
don't think that we are very far from that day.
Yeah, the digital twin is maybe somewhere we are towards heading towards maybe in the coming 10 years, 20 years, where we'll
Right.
have all the data and our digital twin will be a simulated one. And the therapy will also be simulated on that digital twin before applying on us. So it gives how it will actually work on my demographics, my risk factors, my
Right.
genomics. So it is
Right.
the goal maybe towards which we are heading maybe in the coming years.
Mm-hmm. It almost sounds like a big sci-fi kind of a
Yeah,
movie,
yeah,
when we talk
yeah,
about these things
absolutely.
today. About or even talking about such things in most forums. And here we are today discussing these things, and they are already happening. So it's just a matter of time when it will be just, in your backyard where people will be actually employing such tools in their practice.
Absolutely. And now the government of India is focusing on startups. They're giving much funding to the India
Mm.
motion. Everything is a good time actually to explore, collaborate, and do research. Whoever has set
Right.
up, maybe somebody is doing some work in AML. They don't have the tech part, so the tech
Right.
people should collaborate with them. So
Right.
yeah, that's the way forward.
Yeah, I think collaboration is the key word that you have used because everybody doesn't have everything. And it's only that, the people with the like-minded, um, uh, the thoughts or the vision come together and put forward their strengths and, create tools like this. Because otherwise it would be very difficult to get everything under one platform in one place by just a couple of people. So yes, we should keep this forum on even in the future to see whether we could have some synergies, and lead to the creation of a good product which can be used in laboratory practice.
Absolutely.
Are there any more questions, anybody? Lovely thought by Dr. Asta Gupta again. Lovely thought. Thanks again for the wonderful discussion. Yes, it was wonderful interacting with all of you. Definitely, Dr. Utso created a big wave today by bringing up such an important topic across, which made us open our minds. But such a lovely audience as well,
Yeah, yeah.
interacting
Actually,
with
the
us on a
audience
forum
interaction
like
is
this.
more important than the presentation.
So yeah, it goes hand in hand, Dr. Roy. So your presentation was so good that it brought out that interaction. Let me put it that
Hmm.
way. So yes,
No, no.
we would be in touch with all of you. You have our emails. Do write to us in person as well if there's something that we can all work on together. And also do let us know if any of you would like to participate in our webinars also in future. We would love to have you all on our platform as well.
Thank you, ma'am, giving that opportunity today. I hope others will
Anytime.
also explore
My
in the
pleasure.
future. Yeah.
Yes, it was my pleasure, Dr. Roy. Thank you for taking your precious time out on a Saturday evening and being here and enriching our knowledge in the field that you have spoken about. And thank you to all of you who have been on this forum and participating in today's webinar. So if there are no more questions, I think we can call it a day, and then I can wish you all a very happy weekend ahead. So thank you, everyone. We will be posting things on LinkedIn, and we'll share the videos across to everybody who have emailed us. Do keep checking out our space on LinkedIn for more exciting webinars, for more exciting opportunities as well. If anybody wants to come up on our forum and participate, you're more than welcome. Thank you, everyone, Dr. Indranil, Dr. Asta, and the rest of them on the group. We look forward to more of participation and interaction, and wishing you all a wonderful weekend ahead. Thank you again.
Thank you. Good night. Yeah. Good day.